So let us look at what we call chloroplasts. We know that the process of photosynthesis actually occurs in the leaves. Okay, so we also know the internal structure of these leaves, where we look at the upper epidermis and the lower epidermis, and then we look at the mesophyll layers, where we have the pulsed mesophyll layers, the pulsed mesophyll layer, and then also the spongy mesophyll layer. So the arrangement of these layers is not actually the same in terms of how these cells are arranged and packed. Okay, but one key note in that description that you should always remember is the fact that actually in the pulsed mesophyll layer, you have rectangular cells that are having a lot of chloroplasts. Okay, so that means that that kind of arrangement is actually optimal for absorption of light. Okay, maximizing the light that is actually available. But now we want to basically see the chloroplasts which are actually involved in absorption of this light, because in this chloroplast, that's where we have these chlorophyll pigments or photosynthetic pigments that actually trap this light that is used for the process of photosynthesis. So what are the things that are within these chloroplasts? Okay, so how are the things that are found within the chloroplasts related to the process of photosynthesis? So can we relate the structure of the chloroplast to the stages of photosynthesis? And if we can be able to do that, then we shall have understood the concept quite well. So we start with the scenario, and this scenario is about biology students who conducted a study or an experiment comparing spinach plants growing under the shade and those that are in the open, meaning that they have full sunlight. Okay, so they're looking at whether there will be difference in terms of the structure of the chloroplasts. That is, in terms of the distribution of these chloroplasts, whether they are numerous, whether they are few, whether they have some special structures, or the arrangement of the structures. Is it different or is it the same? That's what they are looking at. So of course, they have to use a microscope to be able to look at these chloroplasts in detail, such that they can be able to make the comparison. Okay. So the thing is, of course, for you to be able to make the comparison, you need to be able to know the structure of the chloroplast. So that is the first thing. A typical chloroplast, how does it look like? And in terms of the internal structures, these internal structures, what role do they actually play in the process of photosynthesis? And that is question A. And then question B is that based on the experiment, do you think there will be differences that they will actually notice, whether in terms of size and number of the grana? Will there be anything that you would like expect to see there? And then part C of the question is about how the process of photosynthesis, which has light and light stage and dark stage, or basically light dependent stage and light independent stage. How is the structure of the chloroplast efficient in handling these two processes? Because we actually know that these two stages actually happen within the chloroplast. But what makes the chloroplast to be very efficient in handling these two stages within the same organelle, which is the chloroplast that we are talking about here? Okay, and then the last question is a question to do with can you give reasons why studying chloroplast structure can be useful in choosing crop variety that can grow well in different light conditions? Okay, so that means that if you see the internal structure of leaves, and then you look at the different arrangements of these chloroplasts, it can give you an idea whether this this plant variety is actually coming from a place which has a lot of light, or basically this plant is from a shaded area with no sufficient light conditions. So that means that you can actually be in position to know which crop actually grows where and why, because of the nature of the chloroplasts, their arrangement, distribution, and the internal structures. Okay, so let us look at the chloroplasts in detail and then see whether we can be able to answer the questions that we have in here. So chloroplasts are cell organelles. You can only find them in plant cells and also in the algae, because these are the ones that are actually photosynthetic. You can't find chloroplasts in an animal cell. Okay, so we know that photosynthesis is about making organic compounds, like, for example, glucose, using sunlight energy in presence of carbon dioxide. Of course, you need water and sunlight, right, for this to happen. So basically, the photosynthesis is the process that helps in converting light, sunlight energy into chemical energy, okay, or basically light energy into chemical energy. And that chemical energy is what is found in the sugars, okay, in the sugars. And we have to stress that the process of photosynthesis is actually what every organism on Earth actually depends on. Okay, so that means that photosynthesis is an extremely very important process to sustain life. Okay, and the end result, we talk about glucose as the end result of photosynthesis, but of course, even other organic molecules can actually be made out of this process after after producing these sugars. Okay, so of course you are seeing the structure of the chloroplast there, and we are going to look at it in detail. So we start with what is around the chloroplast. So the outside membrane surrounding this chloroplast is what we call the outer membrane. The chloroplast is actually a double membrane. It is a membrane-bound organelle, right? That means that all organisms that actually have chloroplasts are eukaryotic organisms. So we can say there that we can find membrane-bound organelles in eukaryotic cells. That's some point there. So we are saying that the outer covering is what we call the outer membrane. So is this membrane actually permeable to everything that comes and wants to enter into the chloroplast? The point here is that it is permeable to smaller molecules and ions. That means that the cytoplasm of the cell can exchange content with what is inside the chloroplast, because this outer membrane is not impermeable, but it is permeable to certain molecules and ions. So double membrane. So the next membrane is the inner membrane, and this one, is it permeable? And here we are saying that for it is selectively permeable. Okay, it is selectively permeable. So that is also very important. That means it allows certain ions or particles or molecules to pass through while not allowing others. Okay, so it contains transport proteins. That is actually, it explains why that selectivity is there. When we say it is semi-permeable, or when we say it is selectively permeable, it means that it allows few, not even few, specific because few is different from specific. It allows particular particles to pass through and then does not allow others to pass through. So that definitely means that it is selectively permeable. And the fact that it has transport proteins means it increases that property of being selective, because it is these transport proteins, transport specific molecules. Okay, and then there should be space. So between the inner and outer membrane, we have the intermembrane space. So this space is very narrow, and it acts as a barrier between the cytoplasm of the cell and also the chloroplast interior. So let us talk about the stroma. This is a dense fluid that is found within the chloroplast, and it is surrounded by the inner membrane. Okay, so this actually has a number of things. It has enzymes, it has organic molecules, it has DNA. And now we move to what we call the thylakoid membrane system. Okay, so here we have a number of components, but just to know that the thylakoid membrane system is within the stroma, which is basically the fluid. So it is basically a network of membrane bound compartments that we call the thylakoids. So the thylakoids are arranged in groups which are disc-like. So basically what you see these here, those are the thylakoids. So these thylakoids are arranged in groups, and the groups are the ones that we call the granum. So the whole pack of these thylakoids are the ones that we call the granum. Okay, plural, it is granum. Okay, so we have a lot of them piled, and piled. And then these are actually connected to one pile is connected to another pile. The importance of this kind of arrangement, it increases the surface area for photosynthetic reactions to basically take place. So we say they are membrane bound compartments. Okay, so membrane bound compartments, meaning that we have spaces in them. Okay, so these spaces in them are the ones that we are terming as the thylakoid lumen. So inside here, you see that empty space is the thylakoid lumen. You can call it the thylakoid space. Okay, so this space allows pumping of these of protons, as we shall see later on in the process of photosynthesis, where we deal with protons. So protons basically accumulate during light stage in this lumen, in the thylakoid lumen. And then the surface of this compartment, the thylakoid membranes, is where you have chlorophyll, which is the pigment that absorbs sunlight. Okay, so this thylakoid membrane is the site for one of the stages of photosynthesis, and that stage is basically the light dependent stage, which we basically often call as the light stage. Okay, so we are saying that it is within the membranes of the thylakoid that we find the chlorophyll, which basically captures the sunlight. Okay, and then we said that within the stroma you have a number of organelles, and we can talk about the functions of these different organelles or structures within the chloroplast. Okay, so you have the DNA, meaning that the chloroplast has some degree of independence, that if it requires certain proteins, the instruction can come from the chloroplast DNA. So that means that the other things that are required for photosynthesis, like the ribosomes, are also there in the chloroplast. Okay, so the ribosomes, we have those that are free, And then we have those that are in clusters. Okay, so a cluster of ribosomes translating a single messenger RNA strand, allowing for efficient protein synthesis. When you have ribosomes in groups, that means that they can synthesize proteins actually very fast. So these groups are the ones we call the polysomes. Okay, so we have free ribosomes, and then we also have those that are in clusters. And then other things, we have the lipid droplets, which can be used for synthesizing this membrane and also energy storage. And then starch grains, also for temporary storage of carbohydrates that are produced from the process of photosynthesis. Okay, and then we said that these grana are actually connected using what we call the intergranular lamella. Okay, intergranular lamella, connecting one thylakoid to another thylakoid. Remember, we have the thylakoid being in groups or in piles. Okay, so one granum is connected to another granum through the intergranular lamella. Okay, so the chloroplast, what does it do? We have seen that processes of photosynthesis actually happen within the chloroplast because the chloroplast actually has chlorophyll, which is very important in trapping sunlight. The chloroplast also has DNA and also ribosomes. That means that they can actually synthesize some proteins that they actually need. Okay, so some of these proteins are actually required for the process of photosynthesis. Okay, and then the chloroplast stores some organic compounds that are very important for different processes. So we have the starch granules and also have the lipid droplets, which can be used by the plant as a source of energy. So what makes the chloroplast efficient at the process of photosynthesis, or what makes the chloroplast efficient in photosynthesis? One is that, of course, the chloroplast has membranes, and these membranes are permeable to the things that are actually required for the process of photosynthesis. So things like carbon dioxide and water can enter into the chloroplast, and if things need to be exported out, they can. For example, oxygen and glucose, which are products of photosynthesis, they can be exported out of the chloroplast. And then you have the numerous grana. That means that you have a large surface area for the process of photosynthesis. And then the different pigments that are found within the thylakoid membranes are very important in absorbing sunlight. And then the proteins which are embedded within the grana help in holding photosynthetic systems and chlorophyll molecules in position to be able to capture light quite well. And then within the stroma, you have enzymes for the light independent stage, basically the Calvin cycle. Okay, and then you have DNA within the chloroplast. That means that they can control the process of making certain proteins that they actually need. So we can say that the chloroplast can self-replicate. Presence of ribosomes actually means that they can synthesize the proteins that they need for the process of photosynthesis. So one concept we need to be aware of is the concept to do with compartmentalization. Okay, compartmentalization. What is this concept? That within the chloroplast, you have the two processes or the two stages of photosynthesis, but these processes occur in different sections or in different regions. So there are two distinct regions that we can talk about the chloroplast. So the chloroplast has these distinct regions where you can have the two processes of photosynthesis happening separately. And this physical separation means that you will have different conditions for each of the stages of photosynthesis. So issues to do with pH and ion concentration are actually different in the two regions. So where you have light, the dependent reactions basically happening in the thylakoid membranes, you will have electron transport system, ATP synthase being available. And that means that it is very easy to maintain the proton gradient, the movement of these protons, and maintaining their concentration across these membranes. Very important. And that happens in the thylakoid membrane. So that is where you have the light-dependent stage. And then in the other one, in the other stage of photosynthesis, where you have the light-independent stage, this one happens in the stroma. So within the stroma, that's where carbon dioxide gets fixed and the sugars are synthesized. So that means that within the stroma, you have the enzymes that you actually need for this process. Okay, so that is the compartmentalization that we are talking about. So we go back to our question. So students are studying two spinach plants, and they want to see the nature of the chloroplasts in these spinach plants, which are grown in light and or in presence of adequate light, and then the other one being grown in conditions where there isn't enough light, for example, in the shade. So they want to look at the structures of the chloroplasts and see. Can you describe the main structures of the chloroplasts? I think you can do that, and how they are suited for their function. You should talk about the chlorophyll, you should talk about the grana, you should talk about the enzymes, you should talk about the compartmentalization, and things like that. And then based on the experiment, can you predict or suggest the differences that you could see? For the leaves that are found in the shade, you could expect large grana, few but large grana, because the large grana will mean that you will have a lot of chloroplasts, and that will actually maximize light absorption even in the shade. And then for the other one that we find in direct sunlight, you could expect more grana, but very thin, not very thick. And that again maximize light absorption in those conditions. Okay, and then coming back to chloroplast, yes, the efficiency, the fact that we have compartments in the chloroplast, we have already explained that, and how it is very important because the conditions are not the same. So having a physical barrier is very important. And then yes, chloroplast structure can give us an idea whether the plant variety or crop variety can actually grow well in particular conditions. That actually shows you, for example, if you know that particular plants that have this kind of chloroplasts grow well in shaded areas, if you move these plants or crops into an area of adequate sunlight, then they may not actually grow well. So that's why understanding the structure of the chloroplast actually is very important. Thank you.